project that we've been going on as part of J Clinic, looking at prognostic biomarkers in sepsis. I'd be happy to take questions. I have the chat window up. Stop me at any time. I'll tell you about some of the stuff that we've been working on. I'm going to start with the team here. I'm just the spokesperson, really. It's a collaboration with 4 different groups. The first is that all the work that I'm going to tell you about here has been done by just a remarkable graduate student, Wei Liao, in the group, and I'll put her contact info at the end. She's actually on this call. So I encourage you to reach out to her if you want to collaborate or have suggestions or whatnot. We're also collaborating with Luca Daniels' group in ECS here at MIT, and they have a lot of expertise in robust ML, and specifically his graduate student, Irene, who's been working on this, and his former student, Lily Wang, who's now a faculty member at UCSD. And on the clinical side, we've had a really long-standing productive collaboration with 2 folks over at the Brigham and Women's Hospital, which is Becky Barron and Bruce Levy, and I'll come back to them, but they're really the inspiration for this entire thread of research. So let me talk a little bit about the clinical context here, and it's sepsis. And at the top, I have the definition here, the life-threatening organ dysfunction caused by a dysregulated host response to infection. And I want to unpack that because I think it's important for why we're doing what we're doing. So we all get infections, and then we get sick, and then we recover typically. And that's the ideal response to an infection. You, the infected person, are the host. And what occurs in sepsis is that instead of having that immune response where you fight the bug and then you get better, it becomes dysregulated. And in some sense, sepsis is an overreaction to an infection. When it becomes dysregulated, you can lead to organ dysfunction, and in fact, that can be life-threatening. So that's kind of one way of thinking about it. Sepsis is what happens when the immune system goes awry due to an infection. And in fact, in the clinical setting, killing the bug is basically the first order of business. But even when that occurs and there's no more intruder, you can still be undergoing septic shock and so on. So on the right here, I show you some of the statistics. It's really quite remarkable, the morbidity and mortality associated with sepsis, 10th leading cause of death, $16 billion in the US. And depending on which numbers you look at, really high mortality rates. We think about how scared we are as a society about COVID for good reason. 20, 30 on the left here, depending on which numbers, 50% mortality rates, pretty dramatic. This cartoon on the left has a lot of little nuggets, but the one I want you to focus on here is this 8%. So every hour that sepsis goes undiagnosed, you have an 8% increase in the mortality rate. And the point of that is that as opposed to things that have dynamics of weeks and months, sepsis has dynamics of hours. And so understanding what's going on that hour time frame is actually important. And we also all remember that because 2 years ago when COVID-19 pandemic started, we would see these stories about someone and they would get COVID, and then their blood oxygen would be okay, and then it would crash and they would need to be intubated, put on a ventilator, so on and so forth. So you would get these very fast changes in the clinical state of the patient. And that's actually this thing called ARDS, acute respiratory distress syndrome, ARDS, and that also occurs in sepsis. And so hours are important in the management, in the diagnosis and management of disease, and that'll come back. So there's a lot of needs in sepsis. Of course, people would love to have better drugs and molecules to treat things. But here we're focused on different aspects, and we could think about needs in screening and diagnosis and management. And I just give you just a few snapshots of work in each of those areas, just a lot of work in those areas. So on the screening side, people developing computational tools, scores to be able to triage, for example, high-risk populations that might present at the emergency department and tell you, well, you need to take a closer look at this subset of patients. Diagnosis is actually a challenge itself. So there's people developing experimental tools here where you take an amount of blood and you look at what the immune cells are doing in terms of, in this case, what's known as mRNA expression. And it gives you some indicator that helps you diagnose this faster than you could, for example, by looking at, let's say, blood cultures. And then on the management side, how do you give the right therapy to the right patient at the right time? Here's a paper talking about a reinforcement learning strategy to do this, okay? So lots of work in this area. We're really focused on this latter area of management. It's important to know that there's a lot of different knobs that clinicians can turn when they're managing someone with sepsis, okay? And that's kind of what's shown here. There's different molecules that you can give. Of course, you can put someone on ventilation or not. We've heard about deep sedation in COVID, for example. So there's a lot of different things that you can do. And really the question again is, what do you do for this patient at this time, okay? Okay, and then finally, I wanted to broaden it out a little bit. Here we're really focused on sepsis as the basis of this collaboration that we've had for a long time. But the idea that dysregulation of the immune system is important, actually is important in a wide variety of disorders. This is just a cartoon to illustrate that sort of a different therapy in a different world is in cancer, where people are taking cells from people's bodies, in this case T cells, engineering them to be able to fight the tumor in that patient. What occurs when you infuse these so-called modified T cells back into the patient is you get what's called the cytokine storm. The patient's immune system reacts quite dramatically to the therapy. It may actually be required as part of the therapy, but managing that acute dysregulation of the immune system is actually important in that therapy as well. After having sort of hung out with immunologists for probably too long, you start to get the sense that every disease is an immune disease. That's probably not quite true, but they will certainly try to have you believe it. Okay, so we started working with Bruce and Becky about a decade ago. Again, they're clinicians and they're researchers. They're trying to understand the pathophysiology of sepsis, as well as the immune system, and also they're treating patients. And when they're treating patients, they have access to a lot of information. They have vital signs, heart rate, blood pressure, all of this stuff on a minute-to-minute, second-to-second basis. But the thing that's going awry here, which is the immune system, they actually don't have clear ways of measuring it. And so they came to us, and by us, I mean my lab and the lab of Jung-Yoon Han, who we've been working together with about a decade ago, and said, could you come up with a better way to assess how the immune system is doing? And so we developed a little widget, and the details of the widget don't matter to our discussion here, but it could take a small amount of blood and look at a particular type of immune cell called a neutrophil that I'll get into more later, and give essentially a score, which we call this delta median of IDP. Again, the details don't matter. But over the decade, starting with healthy samples, then going to mouse models, and then to clinical samples, we're able to show that we could, with this score, get a sense of how the immune system is functioning. And here's just some data where each of these data points is from a different patient sample. And we know that, for example, if someone's on mechanical ventilation, they tend to be sicker than if they're not on mechanical ventilation. And this score can distinguish those, where a low score tends to be a sicker individual than a higher score, okay? And so we were working on this, and we came up with this experimental system, and it takes about half an hour to get its data, and it uses, some number of drops of blood. And in that time, the explosion in deep learning came around. And so the question arose, well, could we do better, maybe with a much simpler system, if we incorporated computation in a more fundamental way? And that's really how we got interested in this whole area. And so this example that I just showed you, as well as the ones where I showed you the sort of examples on the screening, diagnosis, and management, they're really, in one case, either developing an experimental system that gives you a piece of data out of it, or taking, for example, electronic health records and adding computation to it. And they're designing each one individually. And really, I guess our hypothesis is, what happens if we co-design everything together? What if we co-design an experimental system that gets us the right type of information out, that when combined with EHR and computation in the right way, does better than each one can do individually? And I'm sort of inspired by the idea of computational imaging, where people have said that instead of making a very complicated microscope, let's say a piece of hardware, getting an image and then processing it with computation, if I can co-design both the hardware and the software, I can either simplify, I can get rid of some of the hardware, or I can do things that the hardware couldn't even do to begin with, right? So can the co-design get us to be able to do new things? That's the main idea. All right, David, go for it.
Before you get into the depth of this co-design process, could you comment a bit about, with regards to the previous slide, how the signal that you're getting from this device, which notably only uses a few drops of blood and runs in minutes, compares to the signal that one would get with a much more traditional, with some of the more traditional assays that are used for sepsis?
Yeah, and we could talk about that for a very long time. The piece of data that it didn't show here, in fact, is how this compares to the most common blood test, which is the complete blood count, the CBC, which I'll get to actually in a little bit later. But the correlation between what we get here and, for example, some of the clinical measures is much better than we get with the CBC. In fact, we did a bunch of different assays using flow cytometry, ELISpot, so on and so forth. And this widget, had the single best correlation to the clinical indicators than any of those.
What about compared to things like lactate?
We didn't even look at some of the laboratory data like that. Bruce and Becky didn't think that, we were doing, I guess, wanted to look at the immune system itself rather than these other indicators. A lot of people have done sort of biomarker discovery based on things that are routinely measured. We're trying to get, what is the actual immune system doing? This thing here is measuring, as I'll get into it later, the activation state of the neutrophils. And I'm not saying that a biomarker panel couldn't do better. As you probably know, there's been an incredible amount of work in that area. Here we wanted to get at what the cells are doing, because the cells are kind of the integrator of chemical signals and then the effectors of the immune response.
And quickly, as an aside, the surviving sepsis guidelines, most updated version in November 2021, actually don't indicate CRP and lactate as a diagnostic or marker of much significance, or procalcitonin for that matter. So white blood cell count's always going to be your SIRS criteria and the way to go.
Yeah, so we're trying to improve upon the CBC basically. And I would say the other thing is that, the CBC is routinely measured only once or twice a day. And so we were trying to get something where, as we think about dynamics of hours, we could eventually get it into a setting where you could measure this much more frequently. Okay, so this is where we'd like to get to. This is the sort of vision slide where you have a patient who's in the sepsis ICU. We have, of course, access to their EHR and everything associated with that. But we would also, at some frequency, either take finger stick samples, or actually they might have a central line and you could actually take blood as often as you want, although small volumes of blood are still important even in those settings. And in just a few minutes with some sort of box, and this is not our box, this is just a box image that I stole off the internet, obtain information that when combined with the EHR would provide some assessment of where the patient is headed given the current trajectory and therapy, and that will allow the clinician to update their treatment. So that's the idea here. And we translate that into what we're thinking about with J-Clinic. We have these kind of two arms where we have learning from EHR data and then learning from blood data, and then combining this at some intermediate state to be able to predict the future clinical state. So I'm going to take each of these one after the other, and I'm going to spend most of our time on the top one here because that's where we've spent most of our time thus far. Okay, so that's our main idea. Okay, so let's talk about the top branch here, and now I have to go back to the CBC, and really the question is, can we develop a simple way to assess immune cell function, much simpler than this little widget that we developed earlier, and in contrast to the complete blood count, which basically takes a census of the blood, we wanted to get what the cells are doing, not just who they are. So because not everyone might be familiar with that, let's look at what's actually in the blood. And so we have our blood, it's about half solids, half liquid. The liquid is the plasma, and there's a lot of important stuff in there, as we just talked about, lactate, as well as cytokines and proteins and things like this. We're not focused on that here. The solids part is where the cells are, and almost all of those cells are the red blood cells or the erythrocytes. The ones we care about are the white blood cells or the leukocytes, and they're at about 1,000-to-1 ratio. And within the leukocytes, we're actually going to focus in on the neutrophils, which in a healthy individual are the most prevalent of the leukocytes. Okay, and so this is what one version of a hematology analyzer would look like that might do a CBC. We want to do something which is not a CBC. We don't want to do a complete blood count. We want to look at the neutrophils in particular and their functional state. And the question is, can we do that from simple cell images? We're not the only ones who've thought of looking at images and inferring things from them. So I'll just give you a couple snapshots here of work in that area. Where here they took a blood smear, which gets stained and gives you cells that look like this, and use deep learning to be able to classify, that this cell here is a myeloblast and so on and so forth. Okay. And on the right, some piece of work which looked at what's known as autofluorescence. So if you just take cells and you don't really do anything to them, but you look at them in a certain type of illumination, they will glow different amounts, which is called autofluorescence, and try to determine whether the T cells, which is a type of immune cell in that sample, are activated or not based on this autofluorescence. So it's an active area of work, but as far as we know, no one's looked to be able to do what I'm going to show you going forward. So we're going to look at neutrophils. Neutrophils are, in some sense, the first line of defense, very important in sepsis. And we're going to look at activation, which is a term which is a little bit fuzzily defined, but we can define it in contrast to being quiescent. So as opposed to in your blood right now, if you're a healthy individual, you have your neutrophils flowing through the blood, those we would call unactivated. And when they get a signal that says they need to do something, they become activated. And one of the things they do is they leave the circulation, they leave the blood flow by becoming sticky and rolling on the lining of the blood vessel called the endothelium, and going through that. They can do a bunch of things. They can release different molecules. They can do this thing called mitosis, where they basically die, but in a way that is helpful for your immune response. one of the things they do is release this thing called ROS, which is a reactive oxygen species. And that will be important because that's what we're actually going to look at here. Okay. And so these are types of neutrophil functions that we want to look at. Okay. And so we asked ourselves, could we just take just plain, very, very simple to acquire images of these cells and actually tell whether they are resting or whether they're activated. And as part of that, we said we should first start by creating very well-defined populations of both activated and unactivated cells. We start with healthy human blood. That's what you typically would do in this case. And we're going to chemically stimulate them. And let me show you how this works. You take blood, you collect it in the Boston area. We can order blood and get it the same day. That comes, let's say, a couple hours after it's been collected. We can, using kind of very simple techniques that I'll show you, isolate out all of the neutrophils, which is, again, this type of white blood cell from this sample. We're going to stain them, and I'll explain why we do that in a second. And then we're going to split them, and we're going to set some of them that we're not going to treat, and then some of them that we are going to treat with this chemical that I'll explain in a second, and then we're going to take pictures. Okay. Now, this is a long time. This is like 1, 2, 3, 4 hours of work. That's not what we're going to be doing in the end. In the end, you would be using a finger stick or a central line, so that takes just a few seconds. In the end, you won't be staining. We're doing this to get good ground truth results. In the end, you wouldn't be chemically activating the cells because this would be from a sick individual that would already be activated. And in the end, we wouldn't be doing our fancy isolation because here we're trying to get very well-defined populations. There's simpler ways of getting, roughly good neutrophil preps. Okay, so we want to get eventually to 5 to 10 minutes, but as a way of getting very well-defined data, we're gonna do a more laborious method. Okay, so looking more in detail at a microscopic level. Yep, David.
Just to provide a bit of broader context, the machines that you showed earlier, for example, the ones that did the CBCs, what's their, so how do they work? Are they also imaging based? Are there other modalities?
Yeah, you can do it a bunch of different ways. You can use flow cytometric type approaches. You can use Coulter principle type approaches. I think it depends on exactly how deep into the differential you want to get. And there are startups that make like benchtop CBC analyzers. You can also just put them on a slide and count things manually, right? So there's a wide variety. Yeah. Okay, so if you look in detail at what's going on in this tube, you start out with blood. And to get out the white cells that we care about, what you basically do is add these molecules and little beads that basically remove everything but the white cells. And so that allows you to get the neutrophils that you care about into this sort of light pink region here. And then we're going to add a molecule which is called the dihydrorhodamine 1,2,3 DHR. And what this does is, if it sees ROS or reactive oxygen species, it'll turn green. Okay, so it'll be an indicator for ROS. And then we incubate with a molecule called PMA, which is a very well-known way of activating these cells. Okay, and PMA will go activate the cells. That'll turn them to cause them to create ROS, reactive oxygen species, which will turn them green. And then we'll just image. Okay. This is what it looks like after you've done your cell isolation. On the left is what's known as a phase contrast image. This is basically the image that you get when you look at it under a standard microscope. These are very easy to acquire, very fast to acquire images. And you can kind of see that there's roughly two populations of objects in here. There's these ones here that have a faint halo ring. These are contaminating red blood cells, or RBCs. And then you have these bright white cells, and these are the neutrophils. Okay. I told you that we went through this kind of fancy cell isolation, but it's not perfect, you have about a 1-to-1 ratio here of the contaminating cells to the ones that you care about. But that's actually pretty good because you started out as a 1,000-to-1 ratio. So you've actually enriched your sample that you care about dramatically. If you look at this under the green channel, the fluorescence, which is also actually a grayscale image, just pseudo-colored here, what you see is that you have green dots. They have some texture associated with them, different brightnesses associated with them. And if you kind of look back and forth, you can see that they're associated with, hello, Regina, the neutrophils. Okay, and again, what we're saying here is that when the cells are activated, that's because they are making ROS. So that activation is related to the concentration of the reactive oxygen species. That in turn gives us a concentration of green molecules, DHR123, which gives us an average green fluorescence intensity. And so these are not linear relationships between these, but these should be monotonic, at least, relationships between these. So brighter green should correlate to a higher degree of activation. That's the idea. Okay. So we take the images, we're going to find individual cells from them. We're going to first do some background correction, illumination correction using very standard approaches, median filters, and so on. We're going to isolate out single cells using watershed filters. And then what we end up with is about 115,000 single cell image pairs. So pairs between the bright field phase image and the associated green fluorescence label. And this is across 15 different healthy donor samples. So we have a biological and biological replicates of 15 here. So if we look at these and we look at our control population, these are untreated, healthy neutrophils. And then our treated cells, these are, again, PMA treated, we think that they're activated. You just pull out randomly single cell images and look at them in detail. I think I can tell the difference between them, and that's a good starting point to be able to train a model to be able to tell the difference between them. These seem smoother with less texture on the inside than the ones that are activated. And that's not surprising if you were to ask a neutrophil biologist. So the first question is, can we train a binary classifier to distinguish between these? We're going to only use the phase contrast images. And so our label in this case is going to be the treatment. So basically, we're going to say that every cell that comes from an untreated sample is itself not activated, and we're going to label that 0. Every cell that comes from the treated sample, we're going to assume is activated. We're going to give that a label of 1. And then we use a pretty standard model based on ResNet 18, pretty standard augmentation, train test split, so on and so forth, even distribution between the two labels. We do a pretty good job of being able to do the classification here in the confusion matrix. You can see that about almost 99% accuracy across the 20-some thousand test images, again, across biological replicates of 15. So we're pretty happy about that. We used GradCam to look at which pixels were important in terms of doing the prediction. And if you look in detail at true negatives, so these are cells that have not been activated and they're labeled or classified as not activated, you can see that the pixels that are important are the ones within the cells. And similarly for the true positives. This makes us happy because it suggests that we're not doing some classification based on some artifact in the images. In fact, even when there's images and that have contaminating objects in them, and you can faintly see another cell over here, you can see that you're not picking up that in terms of what's important for your prediction. So this made us happy, but we'd still like to understand what's going on in the off diagonal, the false positives and false negatives. So we looked into this. And we have a hypothesis, which we haven't proven, but we think it's reasonable, which is that there's issues with how we assign the label. And so let me unpack that a little bit. false negative here is that we have a treated cell that we are classifying and saying is unactivated. And that seems okay. You add this molecule PMA to your sample. It's not necessarily the case that all 100% of the cells in that sample are even competent to be activated. It seems reasonable that maybe 1% of the cells are not competent to be activated. Maybe they're dying, maybe they're in some other state. So that's a possibility. On the false positive side, here we have cells that are from the untreated sample that we classify as activated. And in fact, they kind of have the texture that one might assume is actually associated with being activated. And again, this is not unreasonable also, that in a healthy individual, they might have some cells circulating around that are actually activated. Further, these samples were acquired a couple of hours beforehand. They've been through some processing. Neutrophils are notoriously sensitive to all sorts of handling. So the fact that you could activate some small number of them seems reasonable as well. In fact, we've been working with these samples for a very long time, and it is clearly the case that sometimes we get samples from quote unquote healthy individuals, and those people were not healthy when they gave that blood, and you can tell it in your assay. That's not the case here. We're talking about sort of single-digit percentages, but there's nothing to assume that it's 100% perfect here. So we're pretty happy with the binary classification, so we wanted to move on and do a regression. And so can we train a model to predict the degree of activation? So rather than just activated or not activated, can you get some more resolution within that? And here's where we incorporate the fluorescence label. So the fluorescence can allow us to develop a scalar label, continuous label, based on the amount of ROS, or the amount of green here. So what we do is for each cell, we're going to take a cell mask for the foreground, and that's going to allow us to average the green within the cell. And then as our background, we use a slightly different mask, which allows us to, for example, get rid of contaminating objects in the field of view. And that'll give us the background amount of green in that local area. When we look at this along different cells, you can see that for cells where there's not a lot of green, you get a ROS, which is about 1.0, which means there's nothing above background, so that's good. And then as the green gets brighter and brighter, your ROS label gets higher and higher. Okay, so that makes sense. When we look at the distributions of the labels in our various populations, that also makes sense. So for example, untreated cells, we shouldn't expect many cells to have a lot of reactive oxygen species, and indeed, everything is clustered around one, very tightly distributed. When you now treat these cells with PMA, it spreads out to the right. Most of the cells are in the 1 to 2 range, but we do have some out here in the sort of very high ROS intensity. And we do have some that are very close to 1. So there are cells that don't seem to get activated even under this treatment. When you combine these two, this is the overall distribution. I'll come back to that because that'll be important. Okay, so we trained a regression model similar, also based on ResNet 18, different final layers, of course. Did hyperparameter tuning, ended up using MSE as our loss. And here you can see the training and test during training. And here's what you get. So looking at the predictions versus the ROS label from training and test, and there's some parts that we're happy with, and then there's room for improvement. So let's talk about the parts that we're happy with. As the ground truth label increases, so does our prediction. We see a large peak here. These are the untreated cells, and then these are the treated cells. So this makes us pretty happy. Our percentage error here is about 13, 14%. That's not so bad. And our mean squared error is about 0.1. So that's pretty good when you think about it from this point of view. On the other hand, there's parts that we don't like so much, and that's over here, which is that when our label is very high, our model underpredicts, okay? And so instead of having a unity slope, we see that we have, let's look at the test set here, a slope which is a little bit less than 1. So we tend to underpredict at high activations. We think that that might be due to the imbalanced label distribution. So we don't have a lot of samples up in the 2 to 3 range, whereas most of our samples are down here. And that's also where we do our better predictions. We did look at some of the work that Dina Katabi's group has published recently in iClear, where they looked at this deeply imbalanced regression when you have these uneven distributions, especially when you're trying to predict continuous variables. And we incorporated, and so in that work, they do different smoothing and I think feature space or label space. We incorporated that work. It gives you, a percentage point or so of improvement. We've done other things to do that. It's not clear that the extra complexity is worth the relatively modest improvement in for example, the MAPE. But we think actually the next step is really to go a different way, which is to go to clinical samples. So the other thing I'm not going to talk about is we used conditional GANs to do pixel level prediction. That also ends up being a little bit better than a straight regression. But we really want to focus this work now on going to get clinical samples from our collaborators at the Brigham, and I'll go back to this registry of critical illness in a minute or so. I think there's also a broader opportunity, which is that what we've been doing is a sort of single input-output relationship. We take these cells, we activate them with a single activator molecule called PMA, and we look at a single facet of their response, which is ROS production. But in fact, there's multiple ways of activating these cells, FMLP, LPS, there's receptor-mediated and independent ways of activating the cells. And there's multiple things that the cells can do. They can do netosis, as I show here. They can degranulate, they can phagocytose, they can express different molecules on their surface. And so you could imagine a much more nuanced view of what's going on. And I would love it if you could eventually train a model where from simple images, you could tell what type of activation the cells are undergoing and what caused them to do that. And that would be a really fun place to go. Okay, so if we go back here, I've talked about the top route. Let's talk about the bottom one a little bit. And that's learning the future state from EHR data. We have 2 data sets that we can look at here. So we have this registry of critical illness. This is ultimately what we want to use. This is at the Brigham. This is a long-standing study that's been going on where patients who present at the sepsis intensive care unit can enroll in this study. If they do, blood is taken from them on day 0, which is when they enroll, and then on day 3 and 7 afterward, their EHR data is recorded, and we have access to all of that. The downside is there's a lot fewer records, so hundreds of patients, which makes it harder to train the model. But ultimately, since we want to combine the EHR data with the immune system status from the blood, that's where we want to go. But we also have access, of course, to the MIMIC 4 data, so
can you just talk a bit more about the immune status from the blood? What exactly do you mean here?
Yeah, what I mean is this, Regina, which is being able to get a sense of the function, so the activation of these neutrophils, the sort of some of the mediators of the immune response based on very simple images, a few minutes from a few drops of blood, combining that assessment of the immune system with the EHR data to do better prediction about what's going to happen.
I mean, maybe for this specific, I mean, there are many other measurement of immune system. So obviously it depends on the disease, but how did you come up to, or maybe it's a clinical conclusion that this is the best readout from the immune system?
Yeah, so this came about because this was brought to us by our clinical collaborators wanting to be able to understand better what the cells of the immune system are doing during sepsis. So they wanted to get a functional assessment of what the cells are doing. They thought the neutrophils is the right target to do that. And that's what we've been working on with them. I talked about some other work that was sort of entirely experimental to do that. And now we're trying to see what we can learn from images in terms of what these neutrophils are doing.
I understand this part, but there are other measurements that one of the immune systems that one can take, but maybe for this specific case, this is the best readout to process. Yeah.
Well, we always have the laboratory data. We have what's, which is of course part of the HR. The question here is none of the data that they're getting right now routinely in the clinic actually tells them what these cells are doing. Right? So it might enumerate the cells like the CBC that we've talked about, but it doesn't tell them what they're doing. They can do clinical flow cytometry, but they don't do it because it's too complicated to fit into their workflow. So I think one of the things that we've learned working with them for so long is coming up with things that actually have a chance of fitting into giving them the information that they're looking for and fitting into the workflow that they need. But there's no sense that you ignore or you disregard other things that are other pieces of information that are available to you. Absolutely. So the MIMIC-4 data is a much larger data set, right? So these are patients admitted to the ICU at the Beth Israel over a 12-year period, 39,000 patients. Looking at the sepsis subset from that is still about 18,000 patient records. So this is a lot larger data set, but of course it's retrospective. There's no blood associated with it. So there's only so much that you can do from that point of view. But what we've decided to do is we'll start here, we'll do some training and then transfer over to the registry data. In terms of future clinical state, what we decided to do is use common sepsis clinical scores. There's at least yep, Pete?
Yeah, just a quick question. I thought that MIMIC-4, the one that runs till 2019, actually has something like 380,000 patients.
This is the MIMIC-4 ICU. That might be a subset of MIMIC-4.
Okay, I'm just surprised. I think the number should be larger.
And it may be whether we're talking about patients or whether we're talking about entries or whether we're, we might be talking about slightly different things.
Yeah, okay.
So in terms of clinical scores that are used, there's the Apache 2. We're not going to use that one. We're going to use the SOFA, the Sequential Organ Failure Assessment. This is a score that's a combination of objective things like laboratory measures like bilirubin and so on, and subjective things like this Glasgow Coma score, which gives you a sense of how the nervous system is doing. And basically, you look at these and figure out where you are on, let's say, your bilirubin concentration that associates a number with this. You add up all of those numbers, you get a number between 0 and 24. And that is your SOFA score. A low SOFA score is better, a high SOFA score is worse. And this has a long clinical history. Here's some data from a couple decades ago showing if you look at the maximum SOFA score that someone has while they're in the ICU and their mortality rate, and certainly increasing with that. So it's commonly accepted it has issues, but that's what the clinicians use. And not just the absolute score matters, but also the trends, whether your SOFA score is increasing or decreasing. Okay, so we look inside the MIMIC data set, sorry. We have our static data, we have our dynamic data. There's 87 features in terms of vitals and labs accumulated at different times. So each row will be a different hour. Not everything is measured at every time. Here you see creatinine is measured at time 0 and 7 hours, whereas the blood pressure is measured at every hour. We looked at the duration of stay within this data set because ultimately we want to be able to predict things for a long duration. And so most of the patients, I think luckily for them, leave the ICU pretty quickly. But there's a decent number that stay for 3 days or even 6 days or even longer. We're also, of course, mindful of the challenges, which I won't go into, but also the biases that underlie the data sets, right? These are typically older populations. You know, the people who go to the Brigham may be slightly different cohort than the people who go to Beth Israel, and certainly the people who are in Boston will be different than the people who go elsewhere. So we have to think carefully about that when we think about generalizing and applying anything that we learn here. And so we've started to work on this, and what we're trying to do is to predict the state, the SOFA score, which is our estimate of clinical state, a day, 24 hours in advance. We're using a temporal convolutional network, using causal convolution, so we're only using data up until time t, and you can see how part of that, the convolutional part with the dilation, looks here. I give the archive reference there. And we're just starting to do this, and some of the predictions look interesting. So here, if you look in blue is the SOFA score at any particular time, okay? And in green is what we predict 24 hours ahead. And you can see that there's cases where we seem to be able to predict decreases in the SOFA score that then occur in the actual patient later on. And remember, when we're making the prediction here, we only have data available up to that 40-hour time point. And here you can see where you potentially be able to predict both an increase and a decrease. I could, of course, show you time series data where the prediction doesn't look very good. And so this is the area that we're working on right now. We'd love to talk about that some more. All right, so this is where we're at. I think we're doing pretty good on the classification and regression from the images. So we feel very good about that. We have a really, I think, well-defined data set that we're happy to share. It'll come out when the ISB paper comes out. We're working now on the SOFA prediction from the EHR, and then of course combining those to be able to do the multimodal learning, transferring to the clinical samples. And then again, let me just point out that this is all Wei's work, and she's just remarkable, and I encourage you to reach out to her as well. So with that, Ignacio and Regina, I'll be happy to take any other questions. David.
Thank you very much, Joe. Yeah, I'm sure that there's going to be questions, so please go ahead.
Go for it, David.
First of all, thank you for a really nice explanation, very nice presentation. And I'm very excited by seeing this imaging work as a complement to what's currently available in existing EHR data. Could you talk a bit about how, whether you view the signal that you would get from imaging as independent versus dependent on the existing EHR data? And the reason I'm asking this question is, do you really need to work on this combined modeling versus, let's say, using a much more simple way of combining whatever your best predictor is with the current EHR data and what you're getting from the imaging?
I mean, it's a great question, and it's a little bit maybe a philosophical one, which is that I feel maybe it's religious almost, that if you have access over the entire pipeline of being able to not just deal with whatever data has decided to have been collected because of historical reasons, that you'll actually get better information than you would otherwise. Has that actually been proven out in this particular case? No, because we're doing all either retrospective or observational studies, right? The real point is, can you get to intervention? And that's still a ways off. But that's where we'd like to get to. It is the case that over DARPA and NIH and multiple types of funding, seeing other teams come up with very fancy measures of what's going on in the immune system, Bruce and Becky still say that they don't have what they need. And so, we're following their lead about what they want to measure, and we're helping them measure it. But you're right. I mean, it could turn out that the answer is you add up 3 things that you're already measuring, and that's the best answer.
Oh, I'm sorry. That's not at all what I was
Yeah.
implying. I was suggesting just that this last part might be unnecessary. This last part of this building these convolutional models versus just taking any off-the-shelf method for predicting likelihood of sepsis or various different levels using existing HR data, and using that to guide when to do a intervention to actually push it through the imaging.
Yeah, I mean, obviously we'll be able to run that experiment, right? Because we'll be able to turn off part of the model or not, right? And ask that question. I don't think it can do any worse, and hopefully it will do better, right?
My bet is it won't, but because I think, but I'm curious to hear what others think and why.
I mean, I can't imagine that not knowing, for example, comorbidities, right, has to be important in management of the disease, right?
No, exactly my point. But I'm saying that I think they're really quite independent signals. I think you can basically figure out when to test using the existing data, which would look at comorbidities, and then run it through your assay.
Well, but so the integration then is occurring in the clinician's head, which might be fine.
No, you can pose it as a sequential decision-making problem of when to test, but where the policy is. I don't want to use more time. I'll let other people ask questions.
We should talk about this more though, because this is interesting.
There's a question here, Joe, on the text from Peter.
Maybe Peter can ask it.
Okay. I mean, I can read, but I'd love to hear from Peter.
Yeah, just a little bit noisy in here. The question was just like, when you do this regression task versus the classification task, like operationally, right, in the clinic, how much more useful is the regression task?
Yeah, I mean, I'm always sensitive to not giving too much information, right? It might not have the extra nuance, may not be worth the extra effort. But certainly, first of all, and a point I should have made better, so I thank you for asking it, is that we don't actually care what each individual cell is doing. What we care about is the population is doing. So at some point, we want some sort of summary statistic. And maybe that summary statistic is binary, but it isn't an all or none process, right? And so there is a sense, especially if you're trying to understand trends, that having a continuous value would be of more use than a binary one. But it remains to be seen. And certainly if binary is the way to go, that's what we'll do. I mean, I don't want to do extra complexity for no reason. Sorry, there's one more now in the chat.
Yep.
Oh, that's a really good question. I don't know. We'd have, right. So the sepsis, was it like 2016 or so? There was a change in criteria for sepsis, if I remember that right. It's a really good point. If the standards for management change over the duration that the MIMIC data was acquired, that's I really hadn't even thought of that. So Michael, wherever you are, I can't see on my Zoom screen. Thank you for bringing that up. We'll actually reach out to Roger Mark and think about that. I mean, yeah, sorry.
I just wanted to kind of follow up on Michael's point. One of my PhD students, Adam Yala, actually looked at not on this type of predictors, more standard kind of, clinical feature-based predictors. And what he and the students found out is actually quite surprising using mimic data, that if you're looking at, I don't remember the years that the model was trained, but let's say you train it from 2010 to 2012 data, and this is on 2013, you get a number which is a high number. But as you are, moving forward in time, the performance of the model significantly drops. And they actually look closely with several other students, what changed, and some of it is change of the definition, some of it is change of the tests. There were a lot of things that are changing. So in terms of time, it seems that sepsis model is not doing good survival, which is kind of an interesting discovery because we didn't know about it. And my question to you was slightly different. Let's say, many clinical definition, and not only definition, also the procedures that are how patients are taken care of, change over time, which results in this drop. Do you think that the models, which are kind of more raw data based on raw data, like the one that you are presenting, would survive better over time?
It's a good question, Regina. I don't know that I have something profound to say on that. I'd love to actually meet or put Wei in contact with the student you were just or postdoc you were just talking about, because this is super interesting to think about, especially at this point, right, before you get too wedded to a particular approach. I mean, I have to think that raw data has fewer biases in it. But of course, as you know, if the hospital changes its instrument over time, things, there can be biases, but I don't know.
I mean, for instance, I think the standard of care, I am really not a cardiologist, but I think the standard care changed that, let's say they give them something which changes, let's say, their heart rate or whatever, something changes because the standard changed. So the measurements is the same measurements, but it would be different over time. I think if I remember Adam's numbers correctly, if you're looking like 10-year horizon, it gets close to random. So it's like really, really scary drop. He works on this work actually with a reporter from Stats. So I think when he produces the result, it will be really an eye-opener for the community, but really thinking, how our models survive over time when they're deployed. And I think if maybe I'm not really understanding the deals, but I think this kind of raw data-driven approaches actually have much higher survival chance.
It's something I just didn't think about until you brought it up here, and so I'd love to have a conversation with Roger. Have you had a conversation with Roger Mark about this?
So it's actually he's talking to Leo Celi.
Yeah. Okay.
It's one of the people in that lab to just really understand and explain all these results.
Yeah. No, that's super interesting.
Regina, if I can butt in, I think it's a mixed bag. So, for example, if people in the ICU adopt, let's say, some new medication that tends to drive people's heart rates lower, then even if you're measuring some objective truth, there may still be secular changes in it because of practice. Now, I don't know whether that would apply to Joel's looking at the ROS sensitivity of cells, because maybe those are not subject to secular change because of the way people are being treated, but I don't know enough about sepsis to know for sure.
I totally agree, Peter, with you, but I think that we really need to start thinking when we are thinking, moving forward. Hopefully, it's going to be deployable research. Really thinking about this question, and even if we have several equivalent models that can read you know, produce the same results. All this type of question, how the standard of care in different hospitals will change readout, because if you know the sepsis model that is deployed in many hospitals right now, at a machine learning one, but do we really adjust them for the procedures in that hospital?
I think clinically, as a clinician, in 2016, the updates and changes were actually more to be more sensitive in identifying and diagnosing and recognizing sepsis and classifying as either sepsis to septic shock and removing severe sepsis. So if anything, you probably actually have more enrollment than changes in standards of care. You'd still follow the same interventions. So I actually think it might help you, that revision, in terms of enrollment in the database, clinically speaking.
Regina, what was your output variable or your outcome variable in what you were reporting?
I think that it was actually Adam who did this job, and I would just connect you, Joel, and Adam together to discuss. I am not closely involved in that work, so
That would be fun.
yeah, so you guys can continue the discussion. Okay, so with that, I would really like to thank Joel for a really excellent presentation. Thank you very, very much. And I think there are already some interesting discussion going on. Let's continue the discussion offline. So thank you all, and hope to see you next week. Okay, bye.
Very much.
Thanks,
Bye.
Joel.
Thank you, everyone. Bye-bye now. Thank you.